I think my talk was really divided into three parts. And the first part did give a sort of a broad overview of the NKarta's expansion and engineering approach using a proprietary cell line that stimulates the expansion of NK cells, the transduction of these cells, the engineering of these cells using a gamma retrovirus to program in CAR molecule of our choice, as well as a membrane-bound form of IL-15 to stimulate the ongoing persistence and activity of the NK cells. I gave some early data on the preclinical development of one of our two lead programs, which is NKX019, which is a CD19-directed NK CAR. I provided data speaking to the pharmacokinetics of these cells, speaking to the influence of expansion conditions on the activity and persistence of the cells, as well as some of the advantages of CAR-NKs relative to CAR-Ts, particularly with regard to the inherent cytotoxicity of these cells, so their ability to recognize target cells that may be expressing very low levels of antigen, CD19 obviously in this case, as well as the fact that NK cells, they don't proliferate, they don't expand robustly when they see target cells, and they don't evolve cytokines to nearly the same degree that T cells will, which reduces the risk for cytokine release syndrome. Finally, I talked about a novel program that we work on that combines NK and T cell modalities together. So again, this is using the two separate cell types together as a single product, because what we've observed is that they have synergistic properties. That's the presence of the T cells helps to promote expansion of the NK cells, and the NK cells have, again, a more powerful innate cytotoxicity than the T cells. At the same time, the presence of the NK cells, which are sort of absorbing some of the cytokines produced by the T cells, help to reduce the amount of cytokine that's evolved by the T cells and reduce, again, the risk of CRS. We could use this to dual target the two cell types to get sort of the quick punch of the NK cells with the more kind of durable activity of the T cells. And we've shown both in vitro and in vivo models that these cells synergize very strongly with one another to produce a greater effect than either cell type can alone.